And also I would like to thank him and all the other organizers for calling me here. And this also gives me the opportunity to lift the degeneracy in front of at least a partially biologic audience. Name degeneracy, I mean. Okay, so today I'll be talking about systems, filaments, membranes driven by motor proteins. So in this, during this last few days, you have heard several lectures on motor proteins, filaments, I'm not too sure, but membranes, of course. Let me just, for the sake of completeness, set up the stage. So what we are interested in, let's say if we are interested in understanding a cell, and then it has a big component which is the cellular cytoskeleton, which is made up of several kinds of filaments, associated molecular motors, cross-linkers, etc. And one would like to understand the organization and properties of cytoskeleton and its influence in cell structure and dynamics. Of course, this is the system which also allows the cell to generate motion, generate stress, respond to stress, respond to external stimuli. So cytoskeleton is made up of several kinds of filaments, microtubules, actin filaments, etc. And then associated motor proteins, and there could be cross-linker proteins. Cross-linkers could be non-motor cross-linkers, and of course cross-linking motor proteins can be also there. So for example, this is a picture of these two cells. You can see the cell nuclei. This red shaded region shows actin cytoskeleton, whereas this is the microtubule cytoskeleton. So mostly you see that actin cytoskeleton is sort of the density is higher near the cell periphery, near the cell membrane, which is not shown here. Okay, so these three types of motor proteins, kinesins, dyneins, myosins, families of motor proteins. In that kinesin and dynein families of motor proteins, they move along microtubule tracks. So if one thinks about transport, intracellular transport, then these guys can carry a cargo and move along the tracks and generate intracellular transport. And on the other hand, myosins are associated with actin filaments, and they constitute the actomyosin cytoskeleton in the previous slide, which we showed in this red shaded regions. Okay, so I don't know if they are playing. Okay, so this cartoon animation you must have seen. As a transporter, a motor protein can take a cargo and move along a filament. What you don't see the statistical mechanical aspects, the noise, stochasticity. What you see, it looks like that it's walking like a macro object, like I am walking on this floor. But noise will be there, stochasticity will be there. And also, if you look within a cell, look at certain kind of cargos here, paroxysome, moving along microtubules. So they go forward and backward in some of these trajectories you see, because the microtubules are not all aligned in the same direction. Some are anti-aligned, so they go from the negative side to positive side, whatever. And also, like, if we look at these filaments, filaments are these two types, microtubules, these rigid long filaments, dynamical structures, actin filaments. They also have their own kinds of dynamics, which you heard today in the morning in Nirgov's talk, actin growth, and so we'll be also utilizing a part of that idea in the second part of my talk. Okay, so I'll not get into these details. Microtubules are made up with tubulin dimers, and they preferentially grow in a specific direction. They show dynamic instability, treadmilling, and actin filaments also like grow in from their front by adding actin monomers. And as I was mentioning, these filaments can come together. There could be cross-linking proteins which can make a network of these filaments, which if I just cross-link of this, all these possible cross-linking zones where filaments are crossing each other, then possibly I'll be generating something like a solid. And some of these cross-linkers, of course, can detach, attach and detach, and if they do this turnovers, attachment detachment kinematics, in presence of, let's say, cross-linking molecular motors, which can shear filaments which are antiparallel to each other. In that context, at a later time, this network can start to behave like a fluid. So this is a typical viscoelastic system which behaves like a elastic system over short timescales and typically behaves like a fluid-like system at a long time scale. Timescales much longer than these turnover times. So these are these generic points that in cytoskeleton you have this microtubule cytoskeleton, actin cytoskeleton. They are not really like separated from each other. They are connected. They work in tandem. There is active drive due to molecular motors, let's say. And also this growth shrink can generate active drive, which requires, for example, for microtubule, it requires GTP hydrolysis, it's an active process, and it happens in a directed fashion. So there is a lot of activity in this. The passive cross-linkers can stabilize elastic networks, but they can also undergo turnover, fluidizing them. Motor protein cross-linkers can generate stress in this system, and turnover these cross-linkers, as I mentioned, at long time can generate flow. And this whole active cytoskeleton agent helps the cell to generate transmit stress and respond to external stimuli. So this is the actual problem. Then there could be something very simple one can construct, and we'll be looking at that simpler stuff. So the simple stuff, what one can do is that can take this some of these components of this cytoskeleton. Let's say if I take here we are showing, I think, actin filaments and myosins. So if you just take out actin filaments and myosin, and let's say here in this specific setup, which is known as a motility assay setup, the actin filaments are irreversibly attached. Their tails are irreversibly attached to a substrate, and the heads can move on top of the conjugate filament, here the actin filaments for these myosins. And if they start to move along this, try to move along these filaments, of course they are attached in one end. As a result, what will happen? The filament will move in the other direction. So that generates a system in which this actin myosin motors can generate force on these actin filaments, and then if you go to large densities, it shows some kind of collective behavior, and at small densities also, like each actin filament can show interesting morphological dynamics. So this is one example of such large-scale collective behavior, but this is done with microtubules on a dianine bed. You see that over time they can align and then come into these large vortices, and which are really long-lived vortices. Okay, this also is sort of complex. You have a many-body effect, many filaments coming together and generating this vortex. So one can simplify it further. Ask a question that if I just look at an extremely dilute system, the dynamics I will be seeing is the dynamics of a single filament. So what is that dynamics? Can I understand that? So this is an old work, 1995. So Lipschewer's lab. This was, the experiment was done again with actin filaments on myosin bed. And what they did is to first identify trajectory of each individual filament. So you can find out this filament, do a video microscopy and figure out where their, let's say, the center of mass goes as a function of time. And from that you can plot this, the distance as a function of time curve, and that from that you can get velocities, you can get the velocity distribution in that system, and that's what is done in this paper. Apart from that, you can also do this experiment in which you change the density of myosin motors that coat that bed, increase that density, and ask a question that what happens to the mean velocity. So what you see is that if you increase the myosin density, initially the mean velocity grows, and then it saturates. At large enough densities, it saturates. So this is another behavior you can observe. A third behavior is about the shape of each individual filament. So these filaments are semi-flexible in the sense they have a bending rigidity. Typical persistence lengths are like 17 microns, and they under this molecular motor drive, what you see is that they start to get into this helical shape. And you can also, force the system to get into such helical shape very easily if you just hold one end of that by just generating some defects on the substrate. If they are stuck at one end, then from this other end it comes and then starts to get into this helical shape and keeps on rotating. So that's what was observed. So our first yeah, so that's a, okay, so that's the filament. The filament itself, let's say, it looks like something flexible. Up to some length scale, it looks like a rigid rod. So this is the typical length scale, lambda p, which is of the order of 17 micrometer. So that's the length scale over which, so that's connected to the bending rigidity of this filament. Sorry? So the total length is some 20 micron, that's the total length. So in that sense, if you take the system, this is length stabilized. If you take the system in equilibrium, it will look like a rigid rod. So it's getting into this shape. So we know that it can be bent. It's getting into this shape because of the forces and torques being generated by the molecular motor bed. So it's not infinitely rigid. Yes. So this is the filament itself. I'm showing just the configuration of the filament. So there could be another persistence in a motion. That persistence could be the center of mass itself goes in a specific direction before it changes direction. So we are not talking about that kind of persistence. This is the persistence of the filament. This is the mechanical property of the filament. And showing the, yes. Micron order. So which one? So this one is just actin filament experiment. With microtubule, this is not done. You are asking the diameter of this circle, right? Yeah. Okay. So first small thing one can do is that try to set up a physical model and try to understand if this emerges and if we can understand this phenomenon. So we model this filament as a semi-flexible polymer under some motor activity. We need a model for then a semi-flexible polymer and motors. And then we'll be looking at longitudinal and transverse response to this system. I can put force on top of this and ask a question that how the system responds to it. That's a typical way you probe any system. And we'll assume length stabilized polymer with a constant length, which was done in that experiment. And we'll be first performing stochastic molecular dynamics simulations for simulating the filament, simulating all the molecular motors, which takes time, looks dumb, but started with that, and then do some infield theory to try to understand what happens. Okay, so a little detail about the model, filament model first. The filament we modeled it as a stretchable semi-flexible filament. So filament is made up of these beads. There is some spring and it's stretchable spring, but keeps a average separation R0. So this equation tells you this Bi is a bond vector, and Ti is a unit vector, tangent vector along that bond. And R0 gives you the mean separation, equilibrium mean separation between the beads. So that's the stretching energy. Second part is a bending energy. If you try to bend it, there is a energy cost. So this bending energy cost is given by if you have this local tangent and that local tangent. If there is a strong bending energy cost, they will like to align parallel to each other, Ti plus 1 equal to Ti. If the bending energy cost kappa is small, they will be allowed to move away from each other, and of course there is a competition between this kappa and the thermal fluctuation in the system kBt. Finally, there is an overlap energy. We have chosen something called a WCA potential, but its purpose is basically that if two parts of the filament tries to overlap with each other, so that costs energy. So that they don't do it, it's unphysical, they should not be crossing each other. Twist in 2 dimension, it's not possible. Of course, in 3 dimension, it's possible. So we are just now focusing on this filament moving on top of the substrate. The motion will be looking at the 2-dimensional motion. And also, like, if you just think about this single filament, its diameter is too small with respect to the length. Twist, and if you go really locally, then probably twist plays a role even for the work of the molecular motor. So if it pitches like that, the thread over which it wants to work. Okay, so and then we have to, of course, model the motor proteins. We model the motor protein as active extensible springs with turnover. So what I mean by that, I'll just quickly draw here, will be easier for me to describe. So let's say my motor proteins are irreversibly connected here, and then I have a spring, and then the molecular motor, this is the head of the motor, so it attaches there. And this spring has a typical length scale, I call this length scale Y. So that's the length scale of the spring. So if the molecular motor tries to walk, this spring will stretch, right? So Y will go to some Y prime. So this will stretch if the motor tries to walk. So that's the model of the motor. So motor, if the motor attaches to the filament, then it tries to walk, the spring will stretch. As a result, there will be a force generated on top of the filament. Filament will get a force along this opposite direction of the spring stretching. And the spring can undergo, so there is a attachment and detachment rate. It can undergo attachment via diffusion limited process, that within the capture radius of this molecular motor, if there is a segment of the filament, then it can capture that segment. So it's a so in that sense, it's given by some constants, diffusion constant and the constant separation. So we choose that attachment rate as a result as a constant, and the detachment rate we take as the typical Hill function. So this is, let's say, attachment on, this is off, where detachment rate increases if the stretching is large. So this stretching causes a load, and it will increase as a function of the stretching. So this is in 1-dimensional picture, so this will look like that. So some force scale here, this is a force, some force scale, so that this detachment rate increases if the stretching is large. So if it stretches a lot, it will detach and relax back to an equilibrium position, and then again reattach. So that's the mechanism. And once it's attached, it of course moves in this direction along the filament with a velocity, some active velocity, which depends on a constant active velocity and which also depends on the load experienced by this motor. So if the motor is unloaded, it will easily move with V0. If once it starts to move, there will be stretching, and as a result, the velocity will go down, because the stretching is acting against the load.
Debashish,
Yes.
sorry, one question. That detachment,
Yes.
shouldn't this depend on the exponential of the load divided by kT?
So we are calling this guy as the load. So this is the load force. And then if I multiply it with the delta and kBt, so kBt by delta, I'm calling something like a FD.
So is this small force, small load approximation?
This load, no. So this can stretch and detach.
So the force dependent, I thought it should be exponential, right?
So it's exponential.
I can't see how it's exponential.
This is exponential. Okay, so probably I should write it as. So this is exponential.
Oh, I see. Okay.
This whole thing.
All right.
Yeah, it was not visible.
And the movement is bidirectional, right?
No, once the bead, so that depends on the filament, right? So if the filament, let's say this is, I choose, let's say kinesin. If the kinesin attaches here, it will move towards this. Yeah. There could be a little bit fluctuation on the other direction, which we are ignoring. Yes. Yeah, okay. Next time. Okay. Yeah, sorry. Okay. Okay, so this brings us to, we have to write down what is this expression for the active velocity as a function of this external, as a function of this load generated. So this load, I am saying this load is here, it's written as F. Okay, so towards the center, somewhat center. So that's what, no, so this is not correct. So this is actually the force. So force felt by the, so this does not go with the mod. So here in this equation, this goes as a mod of the force. Okay. So this is a force, and there was this old experiment. So we have taken this data from Srinivas's paper in 2000. So experiment on, and there are several experiments on force response of molecular motors and sometimes of assembly of molecular motors. So this was done on kinesin, velocity as a function of force. These data points are the points taken from this experiment, and this expression is something you have learned about the Michaelis-Menten kinematics. So if you do that Michaelis-Menten kinematics, you'll get this expression for the velocity where this force acts like this external, some load. So load I am saying is here it's generated because the spring stretches, as a result it gets some load. Okay. And these factors, these numbers we get just directly from the fitting with the data. Okay, so we have this expression for this force dependent velocity. So main thing we have to remember that I have a filament can rotate in various directions. So which force matters, that if a molecular motor is trying to walk in a certain direction, the force that matters is in a direction just opposite to it or along it. So we are not taking forces perpendicular to it, perpendicular to the motion. So that's the tangential component of the force. And you take that and then you can write down an equation for the position of this molecular motor. So if I think about this is my molecular motor. So if this filament was static, the molecular motor would move like a given by this active velocity if the filament was static. But the filament is not static, filament itself is dynamic, filament moves by itself. So let's say filament's motion is given by some x dot. So then this should be given by, even if molecular motor I assume doesn't move, it should move along with the filament. So these two terms will then govern the motion of each individual motor protein. And you can, then these things I have already written down. And from that one can think about something called, so this is not necessary for the simulations or developing the model, but it's something important to understand the results. So this is something called the processivity, which gives you the ratio given by the ratio of attachment and detachment rates, and which gives you a measure over the fraction of time the molecular motor spends in an attached state. So probability that it's attached. Okay, so then we put them together and do the dynamics, just to do molecular dynamics simulation of them. Molecular dynamics of the filament in presence of a heat bath and this stochastic motion of the molecular motors. So if you do that, then you'll see that if the activity is extremely low, the filament sort of remains in an open conformation. If you increase the activity, so filament is highly rigid. So in absence of activity, it should remain in an open conformation. If you increase activity, you'll see that at certain value of activity, it starts to get into this helical spiral shape, not helical, spiral shape on 2D. And what we are showing here is that this is a stable spiral over the time span the movie is taken, and the spirals are formed, and spirals can break. So you can start with a spiral, it can break over time and then generates another spiral, and there is no specific directionality. The number of times you get a spiral in this direction, the same number of times you should get a spiral in the opposite direction, because there is no chiral symmetry breaking here. Okay, so that happens, and this is something we are showing, that a time series of a quantity which just counts the number of turns the spiral takes. So if I just plot that as a function of time, so it remains near 0 where the chain is open. At another time point, it can get into a spiral in certain specific direction, which we call positive, a spiral in the opposite direction, which we call negative. And yes. So I'll show some of this, so which allows us also to analyze and predict more things. And yeah, maybe I'll not get into the time scales, but even the emergent time scales you can predict from such analysis. Okay, so what first thing we do is that we want to characterize these phases. We have open state and the spiral state. Characterization is simple. I can just take a probability distribution of this turn number, epsilon, this psi n, and turn number, if the turn number is 0 and picked around 0, some distribution around 0, we call that it's an open chain essentially. But if the turn number is non-zero, then it's of course like in a spiral state. So you see that all these distributions are always symmetric around 0 because there is no symmetry breaking. And then what you see is that, okay, so if the activity, I'm calling something called Peclet, just imagine this is that V0. So if the activity is 0, it doesn't get into spiral state, you get this nice Gaussian shape of this distribution. And then you generate activity, beyond the point you start to see this maxima appearing. So, but what you will see is that as you increase activity beyond the point, this spiral states becomes equally probable as the open state. But if you still keep on increasing that active velocity, what happens is that you again start to lose the strength of the spiral states. So something non-monotonic is happening. You are increasing the active velocity V0. Let's say you can just increase ATP concentration, by which we have shown that the V0 can grow as a function of ATP concentration, let's say from nanomolar to micromolar, and increase the velocity at least 1 order of magnitude, but you can do that. What is happening is that you are trying to push it towards more non-equilibrium state, but it's sort of getting back towards a effectively equilibrium-like state. So what is happening? That's an important question. So quickly, one thing I show here, that if you just plot this point of the maximum, the largest maximum, as a function of this activity, which we call Peclet number. So if you plot it like that, they will see that this remains, the maximum remains at 0 up to some point, and then bigger maximum goes to a non-zero value, and then again it comes back and goes back to 0. So that non-monotonicity is there. So that's one thing, and another thing is that of course there is a coexistence. Okay, so that's the numerical result. Then what you can do is that I see there are this triple maximum, and seeing that I can just immediately know what kind of function it can fit to from our physics background. Several phase transition studies use this kind of free energies. So what it fits to is like psi 6 expansion, and with something that looks like a free energy. And if I just pretend that this expression is, I am getting it from some equilibrium system, then I can predict what is the coexisting densities or coexisting spiral numbers. So I can predict that, then I can predict something called a spinodal instability from this structure and get those plots here. So what you see is that these green lines show this the coexistence lines, and it pretty well can capture the phase coexistence. So there is nothing much to surprise here because the probability distribution is fitted and all these properties are mean properties or the properties of the probability distribution. So there is nothing much to be surprised about this. But there is another part about which is kind of surprising, I will not get into that, is that once I can write down what is the free energy, I can analyze for effective time scales. So I can write down something called a modular dynamics, figure out what are the time scales for relaxation, and look for this psi n statistics relaxation. Psi n as a function of time, that time series, I can look for the relaxation time scales and can compare them. So they also compare well. Okay, that's a side comment, I will not get into that. This is the phase diagram in the attachment detachment versus activity plane. What you see is that for a given attachment detachment value, if I increase activity, it goes from an open state to a spiral state back to an open state, predominantly open, predominantly spiral to predominantly open state. So there is this non-monotonic phase transition. And this phase boundary, the structure of this boundary is sort of simple to predict. What is happening is that the reason this chain gets into a spiral shape is because there is a balance between the torque generated by the molecular motors and the bending rigidity which allows the filament to stabilize against that torque. So this is a torque balance condition. If I just write down this torque balance condition, noting that the force generated here is due to the molecular motors, the total force it can generate. So this is something important here. The total force it can generate is not really given just by the V0. So here, Fa, imagine it's like the gamma V0. So proportional to V0. It's not just given by V0, it also is given by the processivity. So I can keep on increasing V0. If my processivity, let's say, goes to 0, then the total force that the filament will feel will be 0. Then the filament will start to behave like an equilibrium filament. So this processivity one can write down in terms of this and then plug back in here in this equation and you will get, so this Q is nothing but omega 1 by omega 0. So you get this equation for Q with this alpha given by some constants which we don't know from these expressions. So this along with the fitting constant, so this is that dark line.
Debashish, so is it for one filament or multiple filament? There's only one filament.
One filament.
Okay.
So one filament's problem.
But the figure you showed earlier, spiral-like,
yes,
those were for multiple filaments.
No. First thing I showed is the vortex formation. That's many body. And then the second thing I showed is for a single filament on a molecular motor bed. That also gets into the spiral shape. So one can imagine that if one guy gets into a spiral shape, then in a many body situation, several of them going into that, they will compete against each other or may cooperate with each other. So that was the reason for us to study this problem. But turns out that doing multifilament is still is in future. So the issue is that we wanted to do everything with all the molecular motors simulated, which takes a huge amount of computational effort. But okay, so this is the phase boundary which you can, so you understand that this phase boundary is just giving this torque balance condition that if you have at least this amount of torque, only then the filament can get into the spiral. If the torque is less than that, it will not be able to get into that. Okay. But this argument is a pretty mean-field argument. It just assumes that I have a filament which nicely gets into a spiral and never comes out. So one can ask this question that if I now think about a filament being driven by molecular motors, looks like the molecular motors tangentially move along the filament, why cannot I just model it as a tangentially active polymer? So a polymer which just has a tangential activity and moves along that, along its heading direction. So the answer is both yes and no. Number one is that the spiral phase is observed in such active polymer models. But the second point that there is no reentrance with activity. If you keep on increasing activity, it will get into a better and better spiral because there is no intermediate molecular motors there. You are just directly saying that the filament itself is active. Filament from somewhere it's just getting that activity. So this takes us to this general, one general idea is that in many of these biological systems, cell biological systems, when we think about this active, systems are also passive. It's active, it generates stress, but then it has a pathway by which it can relax stress. It can come back to some more relaxed conformation. And if it comes back to that, it's not immediately generating activity. So there is these two states and continuously there is a current, part of the system gets into this state and then comes back. So this keeps on happening. And this keeps on happening both in biological context and also several of our friends who work in active matter, there are also similar things happening. Okay, and the reentrance due to this non-monotonic change in force due to activity, this is basically due to this fact that due to the fact that activity is not just given by V0, it's also given by the processivity. And the processivity itself, if I increase activity, so if I increase activity, what happens is that extension increases, as a result detachment rate increases, as a result overall processivity decreases. So together they generate this kind of profile that you can increase V0, but the force felt by the filament will not monotonically increase, it will increase and then will decrease. So this is not any theory or anything, this is just hand-drawn cartoon. Okay, so this gives us conformations. Then we can ask these questions that what is the response of this system? One response is simple, people have been studying it for a long time, that if I just take this filament as a rigid rod in one dimension, this is studied a lot. For example, this is a very highly cited paper by Stefan Klump and Lipovsky during Stefan's PhD days. Where they looked at this problem from a molecular motor model, which is slightly different from ours. So they didn't have this spring-like object, so which we think is important for certain reasons. Okay, so what you see is that if you take these guys, increase some external load on them, the velocity will decrease and then it drops. So this is the velocity of the filament. So several molecular motors are together driving the filament. Then it happens like that. And if you think about the total number of, so this is the total number of bound molecular motors. So if you look at the total number of bound molecular motors as a function of this external force, that also decreases. And the mean field theory says that will decrease symmetrically, and the numerical data sort of captures, mean field theory sort of captures most of the numerically observed behavior. of this number of attached motors. So how one develops this mean field idea? The idea is this, that what I wrote down earlier as well, the extension increases due to the motion of the filament, due to the active motion of the motor along the filament. Then there is this active force which depends on extension and the total number of molecular motors attached. And then there is this third component, that the filament motion is governed by the active force and whatever external force you put on there. And the number of molecular motors, dynamics of that depends on the attachment rate and detachment rate. So from this you can get it to a steady state approximation and get something at self-consistent equation, y depends on y, and then you can solve it numerically, and these graphs, these lines are those numerical solutions. So from these numerical solutions you can figure out two things. One is that total number of bound motors that one can figure out, and also you can figure out the velocity of this filament, this v. Okay, so with that we come to this, I think I will not be able to get into membranes. With that we come to this last part of this filament system, that if I take this filament system, so this is something long back started from a discussion with the other Debashish Chowdhury in 2008 or 9, and then it took a long time for us to figure out exactly what is happening, and setting up all these simulations. Before we could set it up, we did not know exactly what is happening. The question was this, that I have this filament on the molecular motor bed, and I take this filament, hold this filament at one end and try to pull it up. So peeling the filament from the substrate. The filament, molecular motors are not only walking, they are also grabbing the filament. So you have to really peel it out. So peeling that filament from that substrate. So what kind of dynamics it is, what is the transition? So what we see is that this turns out to be a first order phase transition, in the sense that if you generate force, then when the phase transition happens, that the peeling actually happens, just before that you will get a state in which partially the filament is absorbed, partially it's dissolved, and it's fluctuating strongly between them. And this is something like that order parameter which counts the fraction of the filament absorbed to the substrate, and you see that it remains absorbed until 18 piconewton, and then beyond that it starts to dissolve, and there is a lot of fluctuation at that desorption, and you see this coexisting phase. So I'm showing the fraction of the filament. So this epsilon, the blue line, is the fraction of filament attached, the absorbed, fraction of filament dissolved from that substrate, and this guy tells you the fraction of motor proteins attached. So if the fraction of motor proteins attached becomes large, then the filament is mostly attached to the substrate, and fraction of motor protein attached becomes small, then the filament essentially gets dissolved. So that fluctuation, again, like two-state dynamics you can see here. Okay, and you can figure out the phase boundary between absorption and desorption, and this phase boundary depends on various quantities, whatever controls the properties of molecular motors, including the this attachment ratio, including the bending rigidity of the filament. I can think about actin filament or microtubules. They will not dissolve at exactly the same point. That will happen at different points. For example, for your convenience, I just wrote down these two numbers for actin filaments bending rigidity and microtubules bending rigidity. So this point is like here, that point is like somewhere here, and in this graph we are showing that how, if I just look at actin filament and microtubule and look for their absorption desorption as a function of the processivity, how it changes. So that sort of goes together. But if I think from the perspective of given a processivity, I change the bending rigidity of these filaments. So these are the values of bending rigidity I have given here. These are the bending rigidity of these filaments in water. And you can change salt concentration, the rigidity will change. So there are several ways to even play with the rigidities. Okay, so this transition again can be written down in terms of instability. So what one does is that one can write down, again start from a Hamiltonian of the filament, and this is over small deformations. One can write down this coupled set of equations for the filament's motions. So Z is like the local height of the filament from the substrate, and the number of attached motors Nu. You can write down this coupled equation, then you can look for the fixed point and do a linear stability analysis around that fixed point, do a Fourier transform, and in the Fourier space you will get some matrix and diagonalize that matrix. So I'm just giving you the framework. So diagonalize that matrix and figure out if there is a positive eigenvalue, that means that there is an instability, and where first time this possibly the eigenvalue which could become positive becomes the first time it becomes positive. So that turns out to be this condition, and this condition is something which we have plotted here in all these black lines are that condition in this previous graph. Okay, so we see that this mean field theory with linear stability analysis gives some reasonable prediction about the transition points. Yes. No. So the filament has a fixed length. Yeah. Sorry. Mm-hmm. Yeah. Yeah, right. So that's actually this picture. This red line that you see, that's a fraction of motors which are attached at a given instance of time. All means that fraction of motors which are not participating because that part of the filament has gone out of the reach of that radius over which the molecular motor go and grab the filament. So once it goes beyond that reach, it cannot grab it. So that's how it drops once the filament, you take it out from the substrate. Okay, so then we come to this question that we are saying that, okay, so if I want to do this whole business, I have so many molecular motors on top of which I have this filament, everything one needs to solve, all these equations one need to solve numerically, which is a daunting task. So can I generate a simpler picture for the motion of the filament? So one possibility is that I have this long filament which is persistent. So if I just take a small fraction of it, which I know because of the persistence, if I take a fraction which is smaller than the persistence length, that part will more or less behave like a rigid rod. So even if I know effective dynamics for that rigid rod, if I can write it down, starting from knowing all the parametric details of the molecular motors, then at least up to that length scale, I do not have, so I have a coarse grained picture of dynamics up to that length scale. I do not have to take care of the molecular motors degrees of freedom any further. So with this, from that perspective, so one of the students present here, Chitrak, had done this study. So what is done is that within a persistence length, the filament segment is rigid, then this behavior will be like a straight line moving on top of this substrate. So that means like it's an effectively one-dimensional motion. So it simplifies the analysis as well. So again, what Chitrak had done is that do the full simulation with all the molecular motors of this rigid segment. And what we ask here is that questions about this dynamics. So what is the dynamics? That what we want is that if I can write it down, write the motion of this filament segment as it moves because of some force. So force can have an average part and some fluctuating part. So I can separate them out with an average and the stochastic fluctuation. And then if I know how this average part depends on the molecular motor details, if I know these fluctuations, how it depends on the properties of molecular motors, then I can probably directly use this equation for that segment and don't have to any further go back to the details of molecular motors. Yes. This is the modeling of the, so let's say I have this long filament and then I break it up over length scales which are smaller than the persistence length. Then over that length scale it's like a rigid rod. So I can imagine that I am then stitching several rigid rods together. So first let's try to figure out what is the dynamics of that rigid rod alone. So if I understand that, then what I will say is that this is the dynamics of that rigid fraction. I have to just take the full semi-flexible filament and call each segment is following this dynamics. That's, yeah, in that sense you can consider it a point. That will be probably the easiest thing to do. But yeah, so that's the idea. So, but now still we have a segment in the sense that many molecular motors can attach together, so it's still a many body effect. So what you can do is that you figure out this mean value. So what you see is that this mean value depends on the number of attached motors and the stall force, and the number of attached motors also again depends on the processivity, etc. So you have that expression, and this second expression turns out to have a nice exponential correlation. So this second expression, this delta f, by definition, this is on an average it's 0 because the average part I have taken here. So the rest of the part is just fluctuation that's given by this correlation function, and this correlation function has two quantities given by this strength c and the correlation time tau. So it turns out that this tau is given essentially by the elastic part of this molecular motors. I am not going into the details of this derivation. Yes. From full simulation. So let's say, okay, so I have not told you all these things. So what we are plotting here is that first thing is that mean force. This mean force as a function of the total number of motors that can attach to the filament. So how it behaves. So you see that it grows and then saturates. Second is that all these points are numerical simulation data. Second is that you can calculate this correlation function, right? And you calculate force felt by the filament and the correlation function of that. So you do that, you'll see that they all decay exponentially. So there is a time scale from there. So you figure out that time scale and plot that time scale as a function of the total number of motors present. And you see that as you increase the total number of motors, this time scale also decreases in a particular fashion. And then finally you can also calculate the strength of this noise. So these are all first calculated from direct numerical simulation. And then we are trying to develop a mean field theory giving arguments with the hope that if we have a closed form expression, functional expression for each of these quantities, then I can just start with the molecular motor properties, plug those numbers into this expression, use those expressions in the effective simulation. So that was the idea. So what turns out is that this timescale is essentially governed by, there are several timescales in this problem, but still it's essentially governed by the spring degree of freedom of the molecular motors. Of course, it depends on the total number of motors, their processivity, those are present there, but there is the spring constant sitting here. And this is the correlation time. So the correlation time, so we can analyze all the correlation times, and then we see that this is the time scale. There could be other components, but this is the time scale which is actually governing the behavior. So this for example, this is plotted here. This red line is, this black line is from that. The correlation function, okay, so that works, and from that a couple of things don't work completely. So here first thing is that this the strength of this noise. The strength of this noise, this prediction tells you that this is this black line which fails to capture the values of the strength numerically. but qualitatively it shows the same trend. Another thing it fails to do is that the force it actually generates, the total force generated by the molecular motors, that turns out to be smaller than the mean field prediction. So one can ask a question of why it happens. So I'm giving just answer to the second part, that what leads to this reduction of the total force generated on top of the filament. This is reduced by a self load. The self load is being generated by this mechanism, that molecular motors, if they all work in tandem, they will generate a force in which they will not be competing against each other. But imagine that all the molecular motors are not doing it at the same time. This is stochastic dynamics. So one motor is moving forward, another motor is not. And this motor also has a spring. So by that spring, it's actually acting against the force this other motor could have generated. So there is a competition between them. And these competing forces one can also figure out. So this quantity alpha here is a parameter which is sort of a fitting parameter, but if alpha was equal to 1, then all the motors are moving in synchrony. What we figured out is that if here you see that this graph, this discrepancy goes away, the moment I incorporate an alpha, and the value of alpha that comes out is 0.95. So that's 95% cooperative or moving together, and only 5% of the cases they are not moving together. So that itself generates a competition and a self load, and that value of self load can describe this behavior. And the nice thing is that again for this we have some closed form expression. Okay, so this I'll stop with this partial conclusion. So conclusion from this part is the coexistence of spirals and open chains we see for the conformation of the filament. We studied the transverse and longitudinal response. The transverse response shows a first order desorption. And we have used some effective active motion, developed a picture of effective active motion of the filament segments, and saw that lack of cooperativity generally leads to some self load by the motors. So with that I should stop, but I will need to get to my acknowledgements. Yeah. So this part of study that I presented is essentially done by a previous PhD student, Amit, who is in doing a postdoc in Northwestern University at the moment. And Chitrak has done the other part, who is in the audience. And I could not talk about, and this part of this work was also done in collaboration with Abhishek in IISr Mohali and Abhishek's previous PhD student, Nisha. And what I could not talk about is the work by Shubhadeep, which I said that I'll talk about membranes in presence of actomyosin cortex. So that's the part which we could not have time to get into. Thank you for your attention.
Okay, question, comment?
I'll show. If I draw it here, it becomes easier. So that has this kind of phase boundary. So this part I said that this is spiral. This part I said open, open, but I said predominantly open, predominantly spiral, and predominantly open. In the sense that here in the spiral phase is predominantly spiral. The probability distribution is like that. Open states here, this is predominantly open, this part. And finally here also there is one small region over, it's predominantly open, but before that there was a regime where it was just open. So that's the picture. So this is the attachment detachment ratio versus Peckley phase diagram.
Yeah, so in the last couple of figures, you showed the force, self-load force. So there are actin motors in your model, right? Sorry, not actin, myosin motors for
No, so so all these calculations I have used all the parameters for kinesin.
kinesin. Okay. Like this self-load thing is actually the internal motors like opposing each other. So we can't really say how many motors are actually participating in that, right? Because we had this force plateau
Yeah, so
at one.
from that measure alone, I don't know how many are them competing against each other,
Yeah.
but I know in some sense. I said that
Through the fraction of motors.
yeah, so who are competing against each other is 5%,
Okay.
and 95% are cooperative. So, but it's happening due to all the stochasticity present in this problem. So you can imagine that the kinesin motor comes with its own stochasticity. So in that sense, it depends on the detail, and details are important.
Okay. Also, this is more of a doubt. Maybe it is stupid. In the starting, you had the expressions from for bending and stretching energy, where there was squares of vectors. So I mean, this is just a doubt. So you took the difference of vectors and basically
And
two
then you take
the
square,
magnitude. You took the basically the magnitude of distance and squared it, right?
sort of. So like Hamiltonian is something which is supposed to be a scalar, and
Okay.
this
That was Hamiltonian.
terms are like that. There is a spring kind of term. So what is a spring? In 1D it's easy to see. What is this spring does? So if I have this two guys separated by generic separation xij, so spring degree of freedom, if I write it as xij minus some sigma whole square. So this, if this is an energy term, then this energy term will try to bring the system into a separation xij equal to sigma. Okay, so k by 2. So that's similar order term for bending. So this was stretching, and for bending what happens is that this is bending, right? If it went like that, it's straight, it's not bent. So if this is bending, then what you look for is its local tangential vector. So let's call that Ti plus 1, another local tangential vector Ti. So these two things, if they are parallel to each other, then there should not be any bending energy cost. So the continuum version of this Hamiltonian looks like this. Okay, so this is discretized as something like a Ti plus 1 minus Ti whole square.
Okay, so I have a question. If I understand correctly, when you applied the force on the filament that is absorbed on the substrate and there are motors, so that probably is the force clamp geometry. So a force is applied and you keep it there. But in that context, normally in molecular mechanics, one calculates the lifetime distribution of the attachment. Did you look at that? And by lifetime, I mean when all the n bonds at some point, suddenly all of them are detached. That means they have gone beyond the sphere.
No, we did not. We did not.
Because that actually is a quantity that I think in physical chemistry and even biology, people ask that what is the
Even before
lifetime?
detachment, even before detachment, I mean, if I wait for long enough time, so one possibility, and there could be two things. One possibility is that everyone, whoever was attached, all of them detaches.
Right.
So that's that. I mean, that probability, although will
Spontaneously, even in the absence of force.
even
And then on top of that, if there is a force dependence, how does it,
Right,
the time decreases
so if
with the
you
increase
hold
of-
it at that separation, then what happens? So what we did actually is that we put a constant force and then looked at what happened.
That's the force clamp.
Exactly.
Yeah. The other thing also
The
you
other
can
is
do,
the other ensemble.
force
that I-
ramp.
Separation. I fix the
No,
separation.
another thing is that, your force, you can keep increasing with time.
Yeah.
That is
Yeah,
what's-
force just keeps on increasing as a function of time, and you see what.
Yeah, but then, there one would calculate what is the distribution of the rupture force.
Yeah.
At what force
Right. At
all
what
of them
force
detach.
it breaks. Yeah. So several things are still
Can be
possible.
done, yeah.
Yes.
Okay, so if there is no further question and comment, let us thank Professor Debashish Chowdhury for a very nice talk.